Okay, so this is the second part of the chapter. It deals with how eukaryotic cells reproduce, how they divide. And this is by mitosis. And so here we see a cell, eukaryotic cell, on the left, they're undergoing mitosis, okay, and cytokinesis. So I mentioned last time the big reason eukaryotes can't use binary fission is they're just too different. They have too much DNA. They have multiple chromosomes, not one, and in total hundreds or thousands of times as much DNA. Okay, the DNA is associated with proteins called histones, and the DNA molecules are different. They're linear. And this is actually helpful because now each chromosome has origins of replication, and each chromosome has terminal regions. And so we replicate each chromosome at the same time. Still takes longer because there's so much more DNA. All right, eukaryotes, here's a picture of the karyotype of the human chromosomes. Okay, a karyotype is just a picture of the chromosomes arranged by size. And here, this what we really have is chromosomes are arranged as in pairs. Eukaryotes have pairs of chromosomes. They get half their chromosomes from their mother, half from their father. So we have two versions of the chromosome 1. Okay, chromosome 1 will carry genes for a certain set of traits, and we'll have one version of those genes from our father, one from our mother. So a karyotype shows us this picture. It's stained and arranged according to size. Humans, we spend most of our life as diploid. We have two copies, two sets of chromosomes, 46, 23 pairs, or really 22 pairs, and sex chromosomes, which if you're a female, you'll have a pair of X chromosomes, but if you're male, you'll have one X and one Y. Part of our life cycle is spent as haploid organisms. For us, that's a very small part, just as a sperm or an egg. So eukaryotes have this, and they have these huge chromosomes. Some are hundreds of times longer than a prokaryotic chromosome. Our largest one is 140 million nucleotides. So consequently, it has to be wound around histone proteins. So histones have a charge. They're positive. That attracts the negative DNA. The DNA loop is normally wrapped around there. And so this allows us to fit all this DNA into a small cell space. And we can, normally chromatin is just coiled like this, but when cells are going to reproduce, there's this supercoiling going on. So the DNA molecule becomes this very large visible structure that we can see with a light microscope. The duplicated molecules of DNA are held together by a centromere. There's a certain set of proteins called cohesions that hold this together, and then this thing has microtubules called kinetochores attached to it. Okay, and so chromosomes will have versions of chromosome 1 from each parent, from the father and from the mother. These things, after the DNA has been replicated, will be held together. The duplicated versions will be held together as sister chromatids by the centromere. So you'll have a duplicated version of chromosome 1 that comes from the father, and a duplicated version from the mother. Okay, and this is how we enter cell division. Okay, that those duplicated chromosomes are going to be held together by centromeres at the beginning. And these things are present when the cell is not dividing, which is called interphase, right before that. And during mitosis, those things become the most tightly wound, and then they're going to be separated into two groups. Okay, this picture shows the mitosis in a plant cell. And so 59 is interphase, 60 is prophase, 61 is prometaphase, 62 is metaphase, 63 is anaphase, and then telophase and cytokinesis are 64 there. So it shows the chromosomes being separated, and it's hard to see what's going on there. So let's work with some diagrams. Okay, here, this shows a cell just getting ready to enter prophase. And so when this happens at the end of interphase, the DNA has been replicated, the centrioles have been replicated, and the centrioles are in the cytoplasm, and the DNA is in the nucleus. Okay. During prophase, what happens is the normal cell architecture breaks down, and we start to see the condensed chromosomes becoming visible. And so they're going to appear as two sister chromatids held together at a centromere. The normal things that hold the cell and hold things in position disappears. This is the cytoskeleton, and that's going to be replaced by these migrating centrioles and microtubules that form a spindle between them. The Golgi apparatus, the endoplasmic reticulum, the nuclear envelope are all kind of dispersed and broken down and are going to disappear. In metaphase, what's going to happen is these duplicated chromosomes will line up in the middle, the metaphase plate or the equator of the cell, and the kinetochores of the centromeres become attached to the spindles, the spindle fibers, the microtubules. Okay, here we can see this kind of in a photo through a microscope and through a diagram. The metaphase plate, it's just an imaginary line that forms the center of the cell, but the cells line up on it very precisely. They line up the chromosomes. Half the chromosome kind of pointed one way, half the other way. Kinetochores attached to microtubules. Okay, and this is the trick in mitosis. It's kind of like kindergartners. Line them all up, half go one way, half go the other way. And so this is what happens. And then in anaphase, the proteins that hold the centromeres together, those cohesions, are degraded. And so the duplicated chromosomes are then free, and they're pulled to the opposite ends of the cell by movement of the microtubules. And so you see typically this motion where the centromere goes first. That's because those kinetochores are being pulled that way. Okay, and so this is anaphase. Then in telophase, what's going to happen is those chromosomes are reaching the ends of the poles of the new cells. The nucleus starts to reform around them. And then there's a cleavage furrow, at least in animal cells, where a ring of microtubules will start causing a constriction there, and the cell will pinch. And at this time, other materials will start to reform, the Golgi apparatus, the endoplasmic reticulum. You'll get kind of a random division of things like mitochondria, and we'll wind up with two cells. Okay, here's pictures of this happening in actual cells. And this last part, cytokinesis, is pretty fast, okay, in animal cells. In plant cells, it's a little slower. Plant cells have to form a cell wall, so they have this vesicle forming at the cell plate, and they start depositing cellulose there to form that cell wall. Okay, all right. So that's the short story of cell division in eukaryotes. But the longer story is really something called the cell cycle, which is what we'll go over on Monday. And these are the events that happen in interphase, kind of preparing for cell division and controlling cell division. And so we're going to talk about phases of interphase called G1, S, and G2. And then we're going to talk about the triggers, what triggers things that happen in these different parts of the cell cycle and how it's controlled. Okay, so we're going to stop there. That'll be enough for the weekend. Again, remember, exam is not too far away. Won't be next week, but it'll probably be early the week after.